Hi folks, and welcome to this week's video lesson. We're going to be looking at proteins this week. I'm going to for this lesson, I'm just going to work through the booklet and work through the notes with you on proteins, because I'm then going to give you some practice questions to test your knowledge. So as usual with this topic, you've got the learning intentions here, the learning outcomes for the proteins topic, and we'll just work through the booklet one step at a time. So the first section looks at the role of proteins in the body. So if you guys, I'm sure everyone has heard of proteins. If you're biologists, you might know quite a bit about proteins. But everybody has probably heard of proteins as one of the essential components of our diet. And proteins are also essential for a variety of roles in the body. So the first thing that you need to do in your notes, this is a research task. I'm not going to tell you all of this information because you can find out much more about it online than I can tell you. So we've got a few different proteins listed here. One of them, though, I've got the function, and you've got to match up the name of the protein. So this is a little research task. You just have to research the protein, find out if it's fibrous or globular, and find out the function, and just summarise that in as few words as you possibly can. So what you might want to do is pause the video now and try and complete the table before coming back to this. Okay, so if you've completed the table, then what you'll see is that proteins, like I mentioned, have a whole load of different important roles in the human body. So they're essential structural materials in animal tissue. So our hair, keratin's an important protein, skin, muscle, all of that is all made from proteins. They're also involved in the regulation of processes needed for life. For example, enzymes are proteins which are biological catalysts. So for biologists, this definition will be really familiar, but it's something that's worth remembering for everyone. They control all of the body's chemical reactions, from respiration right through to digestion. So in this little topic in higher, we're focusing mainly on protein structure, and then we're going to go on, once we've understood what the structure of proteins is, we're going to go on and look at the effect of heating on the structure of proteins. So this is how it links into the chemistry of cooking, but it links into a lot of other things as well. So all protein polymers contain the elements carbon, hydrogen, oxygen, and nitrogen. Some also contain sulfur. Now, polymers is a word you hopefully are familiar with from National 5, but we'll explain it in a little bit more detail here. So proteins are polymers, meaning they are made of smaller monomers connected in a chain. The monomers used to make proteins are known as amino acids. So maybe up here somewhere in your notes you can sketch a little diagram. So let's just imagine a chain made up of circles. It can be any shape you want, but the thing to remember is that a polymer is a long chain molecule, and a monomer is the name given to each of those links that make up the chain. Okay, so the monomers used to make proteins are known as amino acids. Amino acids, again, are something that you might have heard of as being really important in the diet. So amino acids are molecules with two functional groups, an amino group, which is an NH2 group, and a carboxyl group, which is the carboxylic acid group, COOH. So the amino acids used in protein formation in living things have both functional groups attached to the same carbon atom. So we've got this middle carbon atom here. It has a hydrogen attached, and then we've got the two different functional groups. So I've asked you to circle and label the amino group and the carboxyl group in this amino acid structure. You can probably do that yourselves, but I'm just going to go ahead and do it. So we've got our carboxyl group here. So remember, our carboxyl group includes the carbon. We've got the carbon double bonded to an oxygen and then an OH, so that's the carboxyl group. And then we've got our amino group over here. Okay, so that's why these are called amino acids. We've got an amino group and an acid group, the carboxyl group. So we've come across this R symbol before. So in an amino acid, we've got these two key functional groups, and then you've got the rest of the molecule, which can be a variety of different things, and we'll look at some examples over the next couple of pages. So these are the building blocks. Amino acids are the building blocks used to make proteins. And proteins are what we call condensation polymers. So at National 5, you might remember that we covered addition polymers. So addition polymers are made when unsaturated, so C double bond C, unsaturated units join. You don't really need to remember that for higher. Whereas proteins are condensation polymers, so they're still made of little individual units connected up in a chain, but the process by which they join together is different. It's called condensation, and you hopefully remember what condensation is already. So there's a wee mistake in the notes here, so I want you to change this. So an amino group, so there should be an O here. An amino group of one molecule links to the carboxyl group of another molecule in a condensation reaction to form an amide link or a peptide bond, or you could also call it a peptide link. And what the process is illustrated just a little bit lower on this page. It's a very similar process to the formation of the ester link. So hopefully you remember condensation is when, what's the definition? Hopefully you're saying two molecules join with the elimination of a smaller molecule. So two molecules join to produce one large molecule with the elimination of a smaller molecule, which can be water. It isn't always, but it can be. So we'll look at the process here, and then we might add a little bit to the notes up here. So we've got our amino acids, and for now we're not going to worry about what the R group is, about what this middle group is, but we've got our amino group and we've got our carboxylic acid group. And just as it shows in the diagram here, our hydrogen from our amino group is going to combine with the OH part of the carboxyl group, and we've got two hydrogens and one oxygen. What do you think that's going to make? That is going to make water. So I've given the game a little bit for question 1 here. And then we get a bond forming between this carbon atom and the nitrogen atom on the neighbouring on the neighbouring amino acid. So amino acids can sometimes be known as peptides, and proteins can sometimes be known as polypeptides. What we get formed is a little bit like the ester link, because we still have this carbon double bond to the oxygen, but instead of an O, as in the ester link, this is an NH, and this is called the peptide link or the amide link. So proteins can be known as polypeptides. They can also be known as polyamides. And if we remember that poly means many, then polyamide or polypeptide just means that we've got lots of these peptide or amide links in the protein molecule. I think you often see polyamide in the context of proteins as materials, whereas polypeptide would maybe be more common in a biology sense, possibly. Okay, and then for question 2, you've got to write the symbols for the 4 elements present in all proteins. So you should know the 4 elements present in all proteins. It says it at the top of the page as well. Okay. So I'm not going to fill that in for you. Well, actually I will, just to make sure we've got our notes correct. So all proteins contain carbon, nitrogen, oxygen, and hydrogen. Okay, so a question type that might come up when it comes to proteins is you might be given the structure of different amino acids. Usually, quite often it's 3 amino acids that they give you, and you have to draw the molecule formed by the condensation of the 3 amino acids. So most proteins are made of more than 3 amino acids, but you can just get small, they wouldn't really be called proteins, they would be called tripeptides. You can get smaller molecules formed by condensation of amino acids as well. So we've got 3 examples of amino acids here: glycine, alanine and valine. There are about 20 amino acids in total which make up all of the proteins in the human body. And you can see that all of these 3 amino acids, they have the amino group, the carboxyl group, and the only difference is in one of these what we call substituents around the carbon atom. So you've got your carbon atom, and you could say that it's got 4 different substituents. Okay, so probably the easiest way to do this when you've got your 3 structures here, it might be worth circling the atoms that are going to be removed to form the water molecules in the condensation reaction. So this has been drawn in such a way that it makes it reasonably easy. So it's the OH from the carboxyl group combining with the H of the amino group on a neighbouring molecule. So we've got one there and one there. So these are going to be removed. And then we're going to end up with 2 amide links. So for me, it's probably easiest to draw the amide links first. Or at least draw the first amide link first and then fill in the rest of the molecule. So we've got our amide link here. You can, just like with the ester link, you can picture a dotted line parallel with the carbon double bond oxygen and between the carbon and the nitrogen that shows you where you would split this link back up if you're wanting to go back to the original molecules. So our glycine on the left had a carbon with just 2 hydrogens on it. And then the amino group. Now notice that because we are only combining 3 amino acids, this end amino group just remains the same. There's not any condensation reaction taking place here. And then we've got our alanine in the middle. So that again has a hydrogen and a CH3. So take care when you're doing these, if you're just writing CH3, that the bond goes from carbon to carbon, not from carbon to hydrogen or into the middle layer. And then again, we've got our water, a small molecule being removed in the condensation reaction, and we've got this amide link or peptide link being formed. So we'll just draw that in. And then we can draw the rest of our valine. So valine is a little bit more complex. So we've got our carboxyl group at the end. And again, because we're just asked to draw the tripeptide, we're not having any other reactions at the ends here because we've just got 3 joining together. So we can write OH or we can draw it out in full. And then again, taking care that the bond goes from carbon to carbon, not to this hydrogen, because it's the carbon that is actually taking part in the bond. And that's why we write H3C on the left hand side here instead of CH3, otherwise the bond would go from carbon to hydrogen, and then again CH3. Okay, so hopefully that makes sense and you'll get a little bit of practice in the questions. So just as a little recap, what we've learned so far about the structure of proteins is that proteins are condensation polymers and they are made from monomers known as amino acids. So that's the basic structure of proteins, and what we're going to look at now is we're going to look at the what we would call the sort of 3D structure of proteins. So typically we get 200 to 300 amino acid molecules condensing to form a protein molecule. And I find it amazing that all of the proteins in the human body are made from just 20 amino acids. One interesting and important point is that the body cannot make all of these amino acids. So the enzymes can use chemical reactions in the body to make quite a few of these amino acids from scratch, from other materials, but around 9 of them are known as essential amino acids, and these must be acquired from the diet. So there are certain foods which contain these essential amino acids, and they are our only source of certain amino acids that we need to make the proteins in our body. This term comes up a lot, so make sure you know that essential amino acids are amino acids which must be acquired from the diet. Okay, so we've only got 20 amino acids, but we've got a huge variety of proteins, and you did a bit of research of that into that on the previous page. And what creates the huge variety in proteins is the really it's the sequence in which the amino acids are connected. So there are only 20, but if you can imagine 20 different things, you have 200 of them in a row. There are almost limitless different possible combinations, different variations of order that the amino acids can join together in. And this allows the proteins to form an array of complex shapes, including spirals, which can also be called helices, and sheets. So we're not going to look, looks reasonably complicated. We're not going to go into the detail of how this happens, but it's important for you to know that proteins can form complex 3D structures. So one protein, one chain of amino acids might form a spiral or a sheet that might then tangle itself up a little bit more to form what we call a tertiary protein structure. And then we can actually get two different chains of amino acids combining to form what we call a quaternary protein structure. You don't need to know these different names, so you don't need to know the primary, secondary, etc. This is the key bit. The key bit relating to chemistry as well is that these complex 3D shapes are held together mainly through hydrogen bonding. So if we look back to just the general structure of a protein, we can see that we've got in fact where will I draw it? I'll maybe just draw it over here. So I'll draw out the amide link again, and in our amide link we've got the NH, which can form a hydrogen bond because our nitrogen is going to be negatively charged and our hydrogen is going to be positively charged, or slightly positively charged rather, slightly negative and slightly positive, so it's a highly polar bond. And then our carbonyl group as well, the oxygen in the carbonyl group is going to be negatively charged, and this is quite a polar group as well, and we can get a hydrogen bond forming between the hydrogen of an NH or OH group and the oxygen of a carbonyl group. Okay, so main thing to remember is that hydrogen bonding is important for the 3D structure of proteins. Okay, so we've looked at the 3D structure of proteins and how proteins are made, and now we're going to look at the digestion / hydrolysis of proteins. So hydrolysis is the chemical reaction that breaks down proteins, and this happens during digestion. So digestion always involves breaking down of the molecules in food. And we have to break down our molecules of food so that they can be absorbed through the gut wall. So large molecules can't be absorbed into the bloodstream. So digestion, in digestion we need to break down any large molecules like proteins, which are made of lots of small parts, into smaller pieces so that they can be absorbed by the body. So proteins obtained by eating plants or animals are broken up during digestion by a process called hydrolysis. And you should remember what hydrolysis involves to produce amino acids. So again, we've seen hydrolysis in the context of esters, and we're going to look at it in the context of proteins now. So hydrolysis, remember, is splitting using water. Literally it means water splitting, but we're splitting up a molecule using water. So again, we're just going to use a tripeptide as an example, obviously, and like we've said, most proteins would have a couple hundred amino acids connected in a chain, but we're just going to look at one with 3. Now, first of all, how can I tell that there are 3 amino acids in this? Well, I'm looking out for the amide links or the peptide links. So if I just circle these, I've got 2 amide links here. And if I've got 2 amide links, then I've got 1, 2, 3 amino acids altogether. And just like in esters, when we're splitting up a protein chain, or when we're splitting this amide link, we can think about it as splitting along a dotted line parallel to the carbon double bonded to the oxygen, and between the carbon and the nitrogen. So I find it helps me to draw that dotted line in here. What's then happening is then just the reverse of the process before. So our water is going to split as well. In fact, here I'll draw this one like this. Okay, so our water molecule is going to split as well. The OH part of the water molecule is going to join back onto the carbon to regenerate the hydroxyl group. So remember, we removed the OH from the carboxyl group before when we do condensation of the amino acids. So to regenerate the amino acids, we've got to add the OH back on to the carbon with a double bond to the oxygen, and this H is going to add back on to the nitrogen to regenerate the different amino acids. So you can add these arrows in just to kind of help you if that helps you to remember what happens in this hydrolysis. So again, we'll do some more practice of this in questions. And again, we've just got some more examples of some of the amino acids that are used in the human body. So we've got aspartic acid. So aspartic acid is an amino acid, but it actually also has another carboxyl group up here. Serine, which is an amino acid with a hydroxyl group, and then threonine, which is an amino acid which also has a hydroxyl group up here. Okay, so a wee bit of practice for you now. Pause the video now and see if you can draw the structures of the 3 amino acids formed on hydrolysis of this protein here. Okay, so if you're watching this, hopefully you've given this question a go already. Okay, so here's your answer here. So just like in the previous example, we're splitting this chain. You can think of it about as drawing a dotted line parallel to the carbon double bonded to the oxygen, and then regenerating. Every time there's a carbon double bond to the oxygen, that's regenerating into a carboxyl group, and every time we've got this nitrogen here, that's regenerating, getting an extra hydrogen back, and ending up with as an amino group. Now, something you might not have been sure about was what to do at these ends here. So these ends just imply that the chain continues. So what we would call this a free bond, and we don't often draw a bond without anything attached to it in chemistry. But one case in which we do that is with polymers. So these free bonds imply that the chain continues, but because this question is asked to draw the amino acids formed on hydrolysis of the protein, we're going to assume that that there was a peptide link here, that it just continued, and that we would regenerate the carboxyl group where we have the carbon double bonded to the oxygen, and the amino group where we have the nitrogen bonded to hydrogen. I think an exam question would make it really explicit though, so don't worry too much about that. But as long as you were able to draw the basics of these structures, especially the middle one, then that's great. Okay, so we've looked at how proteins are formed from amino acids via condensation reactions. We've looked at the 3D structure of proteins. We've looked at the hydrolysis of proteins. And the final thing we're looking at here is heating proteins. Now, I'm not going to read through all this information in the video. I'll just read through this top box here, and then I've got a couple of little notes I want you to add in this section. So when a protein is heated, intermolecular bonds are broken. So these are the hydrogen bonds. And the hydrogen bonds break before the covalent bonds, because hydrogen bonds, whilst relatively strong, are still much weaker than covalent bonds. And the protein structure, when the bonds break, the protein structure is irreversibly changed. So quite often what happens is the hydrogen bonds that are holding it in shape break, but then new hydrogen bonds form which hold it in a different shape. And when these permanent changes occur, a protein is said to be denatured. So that's the key word that we use to describe permanent changes to a protein structure caused mainly by heat. So like I say, you can have time to read through this yourselves. So there's a couple of things I want to just make clear. So denaturing, heating protein, changes the texture of foods, and more detail about that is given in this section here. So thank you for listening, and as usual, please get in touch with me if you have any questions at all on this topic, particularly if you're stuck on any of the questions.